So I'm guessing everybody is now in the presentation. So maybe if I can just quickly run through the sort of seminar etiquette, and then I can introduce our speaker. So again, if everyone can keep their mics switched off during the talk, that will avoid any background or any feedback, and keep your videos switched off during the talk as well, please. If you want to ask a question, probably after the seminar, then put your hand up that you can do in Zoom, and then when it comes to your turn to ask the question, do switch on your video and mic. But equally, you're more than welcome to type questions in the chat box, and we will do our best to read those out to you and to get to those. And then just to say, if anyone has any further comments, suggestions, or questions, then they can send an email in either to Anne-Marie Minihane, the Norwich Institute for Healthy Aging Director, or a general email there as well. And we can be communicated with via Twitter or X or whatever we're calling it these days, and our website. So please feel free to make use of those. So if I stop sharing now, and now I can kind of see people again, hopefully. So again, it's an absolute pleasure to chair this NIHA seminar and to introduce our guest speaker, Professor Julie Lovegrove, for everyone. And even more so, really, because we tried to do this in July, but a failure of technology led to that not taking place. And Julie has very kindly agreed to come back and give it another go, which is a brave and kind. So thanks, Julie. So Julie did her BSc at Surrey University on human nutrition and dietetics, and before pursuing her PhD studies there on infant milk allergy. And she developed research interests around nutrition and cardiovascular disease risk as a postdoc. In 1996, which doesn't seem too long ago to me, that's for sure, she moved to Reading and the Hugh Sinclair Unit of Human Nutrition. And she was integral and instrumental in developing Reading as a centre of excellence in nutrition research. And she now heads that unit. And so Julie has a very strong international profile in the field and many roles in the area of nutrition. So to give you just a few, she sits on government advisory panels like the Scientific Advisory Committee for Nutrition. She chairs an ILSI, International Life Sciences Institute Committee. She's fellow of the Association for Nutrition. She's just, I think, come to the end of an extended term as president of the Nutrition Society. And in 2022, just last year, she won the British Nutrition Foundation Prize for her outstanding contribution to nutritional science. So all fantastic accolades and roles. She has clearly hundreds of very high quality scientific publications and a great media presence. And her research interests are really around the role of nutrition in cardiovascular disease risk, but also wider in sort of metabolic syndrome and type 2 diabetes. And that's the influence of dietary fats, as well as plant-based diets and phytochemicals, which she's going to talk to us about today, encompassing things like gene-nutrient interactions and sort of personalized nutrition. So today, it's a great pleasure to introduce Julie to talk to us about living on the veg, the impact of plant-based diets on cardiovascular health. So thanks very much, Julie.
Thank you very much, Ian, for that great introduction, and thanks, Anne-Marie, for inviting me here. I'm delighted to be here and glad that I'm able to join you. I'm just going to try and share my screen. Hopefully this works. Can everyone see that?
Yes, we can see that.
Lovely. I'll make a start. So thanks very much for joining us. I'm going to talk to you today about the impact of plant-based diets on cardiovascular health and really cover some of the work that's been done at the University of Reading within my research group over quite a few number of years. I don't need to tell this audience that cardiovascular diseases are the biggest killer in the world. And you can see from this map here, with the darker colours being higher prevalence, that actually it's much more of an Eastern disease now than a Western disease when I started my career. Despite that, every 8 minutes within the UK, someone dies from coronary heart disease. So it's a really important killer within the UK and more than worthy of research into how we can reduce its risk. This data here shows the impact of diet and its attribution to deaths globally. And if you look at the top 9 contributors of diet, you can see 6 of these are related to plant-based diets, with higher intake of components of plant-based diets related to a reduction in risk. And a lot of this risk is associated with this dark color here, which is cardiovascular disease. Of course, for this reason, we have a number of different dietary recommendations. I've put up here the Eat Well Guide, which was 2016, and you can see the green area of the plate giving the greatest proportion of our diet should be from fruits and vegetables and plant-based foods. Unfortunately, as a population, we're not so good at complying with these recommendations. And this is some data here published a couple of years ago, and showing that only a quarter of the population comply with our fruit and veg recommendations, which is 5 portions of a varied number of fruits and vegetables a day. Around the world, there are a number of recommendations for fruits and vegetables, and you can see within Europe, they're generally about 5 portions a day. In Greece, it's up to 9, and other countries have different amounts, with the WHO saying it should be about 400 grams a day. In the Netherlands, they actually define that you should have equal quantities of fruits and vegetables. But it isn't really known whether there's any particular fruits and vegetables that have particularly important impact on risk reduction, and that's something that we have addressed within our research group, as I know you have in UEA. So fruits and vegetables contain a large number of different, not only nutrients, including micronutrients, fiber, vitamins, but also compounds that aren't particularly nutrients, don't have a deficiency associated with them. And we're particularly interested in flavonoids, and these are found quite high in quite high amounts of fruits and vegetables, and often cause the colour in these compounds. There's different classes of flavonoids, and you can see here a number that are associated with different types of foods. So you have flavanones, which are citrus related, flavanthriols in particularly in cocoa and also tea. And if you look at the contribution that these, or the intake of these throughout Europe here, you can see they vary with country. And I was quite surprised when I saw this, that actually in Ireland and in the UK, we have quite a lot of these flavonoids, but they're mainly from this grey area, which are 3 are flavins and 3 are rubins, which are from tea. So you can see that we are quite high tea consumers within Ireland and UK, no surprise, I'm sure, to most. If you look down in Italy, you can actually see that the majority of their flavonoids are actually proanthocyanins. So it does vary around Europe. So this is quite a complex diagram here. I'll try and take you through it. It's looking at the metabolism of flavonoids. So if you go from the mouth where we consume flavonoids, when you get down to the stomach, you get conversion of oligomeric to monomeric compounds. In the small intestine, you get hydrolysis of glycosides, methylation, and also sulfuration. And then those that aren't absorbed, about 20% are absorbed, and further metabolized in the liver, where you get methylation and other metabolism. Those, about 80%, we think go down to the large intestine, where they're biotransformed by the gut microbiota. About 70% of this is estimated to be absorbed and again metabolized in the liver, and then finally excreted in the urine. And then the 10% is excreted in our feces. And this does vary depending on the type of flavonoid. So really the body treats them as toxins. It's trying to ensure that they are soluble in aqueous solution and are excreted in the urine finally. But we find that these, when present in the body, do have beneficial effects. And the time course of these very much varies, whether they're from the gut transformation or from the stomach, for example. There's been reports of a number of potential health benefits of flavonoids, and these include the brain, cancer, inflammation, but also vascular function and cardiovascular risk reduction. And it's really this area that I want to concentrate on my talk today. So there's been a recent publication just last year of this very impressive Cosmos study, which was funded by industry, and it's one of the only studies that actually looks at the impact of a flavonoid, cocoa flavanthriols. This was a supplement in a large number of people, over 21,000 adults in the US, with a 3.6-year follow-up on hard clinical outcomes. And what was found in this study was that there was a significant reduction in a number of cardiovascular events, particular deaths in cardiovascular disease, with overall outcome of a significant reduction in the realms of about 70 or a reduction of about 21%. So this is quite important. And these levels, although in the form of supplements, possibly could be given or taken as a diet, perhaps a couple of cups of tea and an apple or two a day would give us about 80 mg of this epicatechin. So how does this have an effect? We know that it's really important in cardiovascular disease to have a healthy endothelium, and these are a diagrammatic of the cells lining our vasculature. And this is impacted by many different factors, include smoking, aging, menopausal status, but also diet is really important on the health of the endothelium. If you get endothelial dysfunction, if you're a smoker as we get old, then you can get a number of dysfunctions such as reduced endothelial nitric oxide activity. This enzyme produces nitric oxide. And nitric oxide is one of the most important vasodilators, so allowing our blood vessels to dilate. If you get this endothelial dysfunction, this can lead to atherosclerotic lesions and also finally to cardiovascular disease events. So we want to find ways in which we can increase the health of our endothelium. So this is a study we performed a number of years ago, published actually 10 years ago by a PhD student of mine, Trevor George. It was a small study, but what we wanted to look at is the impact of a flavonoid-rich juice there postprandially, so just over a period of a number of hours. And we looked at here at the microvascular reactivity, which we measured by laser Doppler imaging. So you can see here our sugar and energy match control had no effect on our endothelial dependent vasodilation. But then after the consumption of the flavonoid-rich juice, again, this was a crossover study, we found a significant increase in the vasodilation. This was also associated with an increase in circulating vanillic acid, which is a flavonoid that's through the first metabolism. But also we found excretion of urinary hippuric acid, which is a later excreta and is really reflective of colonic metabolism. So this double peak we saw in benefits in relation to microvascular reactivity could be due to initially the primary metabolites, but also possibly those that come from the colon. Another study that we performed, which was a much larger study that Anne-Marie was involved with when she was at Reading, was called the Flavor Study. And what we did here is we increased the consumption in a number of participants, 150 participants, that were randomly assigned to either a group that was low, sort of a control group that just consume their healthy, consume their habitual diet. Then we had a diet that had low flavonoid fruits and vegetables, and we increased the portions by 2, by 4, and by 6, as you can see over an 18-week period of low flavonoid fruits and vegetables. And then in the high flavonoid fruits and vegetables given here in red, we increased high flavonoid fruits and vegetables by 2, 4, and 6 portions over the 18-week period. And you can see here quite clearly that this particular colonic metabolite of catechin here was increased after 6, 12, and plateauing there at 12 weeks. And we also found that there was a significant increase in microvascular reactivity in the high flavonoid group, which again plateaued at about 12 weeks, with a reduction in and TNF-alpha, a cytokine, in the high flavonoid group. So we showed here in this study that the fruits and vegetables were beneficial, but those that were rich in flavonoids, as opposed to those weren't, had a greater benefit on vascular reactivity and some implications of cytokines. Flavon-3-ol, as we've seen before in the Cosmos study, has been associated with benefits in relation to cardiovascular risk and also vascular reactivity. And this is a paper here from Lee Hooper. Obviously, you know Lee very well. And what she has reported in this is from randomly controlled trials and meta-analysis showing both acutely, so postprandially, and also chronically after 2 to 18 weeks, there's a benefit, a significant benefit of flavon-3-ol on vascular reactivity. And this was measured in this particular study by flow-mediated dilation, which is looking at the dilation of the brachial artery with high levels being beneficial to health. So a study performed at the University of Reading by Jeremy Spencer's group was interested to look at cocoa-derived flavonols, so again, flavanols, in relation to not only vascular reactivity, but also to the impact on the gut microbiome. So in this study, there was 22 healthy men and women. They were randomly assigned to consume either first high cocoa flavanol, which is about 500 mg a day, or low cocoa flavanol for 4 weeks, and then they were crossed over. And this intervention led to the high flavonoids having a significant reduction in triacylglycerol postprandially and also C-reactive protein. And what they also found here is in the high flavonoid group, which is the grey boxes, there was a significant prebiotic effect, beneficially increasing the bifidobacteria and the lactobacillus bacteria after the high flavonoid supplement, and a significant reduction in Clostridia. So you can see that the flavonoids were metabolized by the particular bacteria here that are known to be beneficial. And it could be that this gut microbiome metabolism may be a crucial step in the beneficial effects of flavonols. We're also interested to know that whether there's any impact on genetic variation or genetic SNPs, polymorphisms within an individual. And you can see from all of my slides and others that the variation in individuals is very large. We know a particularly important gene here, the endothelial nitric oxide synthase involved in nitric oxide synthesis, has a SNP that is quite common within the UK population, with about half having the high risk and half having the wild type. And you can see here that those carrying the wild type of the risk variant of eNOS shows with an increased risk of coronary heart disease of the odds ratio of about 26% higher. So we were interested to look at those that had this particular variant, whether they responded differently to our flavonoid-rich juice. So this is going back to Trevor George's work. We split our population. It was only a small population to begin with, but to those that were wild type, so the GG variant, and looked at their response. So we're going back to the endothelial-dependent vasodilation, no impact of our control, but a significant increase in endothelial-dependent vasodilation in this wild type after the flavonoid-rich juice. When we looked at the GT risk variant, we found that they did not respond to the flavonoid intervention in relation to impacts on endothelial-dependent vasodilation. So they had no benefit of this. And we also found that those in the GG variant, the wild type that had the beneficial effect of flavonoid, also had lower LDL oxidation, which is again a risk factor for cardiovascular disease. So this needs to be repeated. It's a very small study, but does indicate that there may be differences in responses depending on your genotype to which fruits and vegetables may be beneficial to an individual. So there's a number of possible mechanisms that could occur, and I have alluded and shown you data on some. And this is a very brief overview of potential mechanisms. So they can be linked to reduction in blood pressure, I've shown you, involved with improvement in endothelial function, also related to platelet aggregation, a reduction in that, and it has anti-inflammatory and perhaps enzyme modulatory effects, and overall has a beneficial impact on the vascular function. Flavonoids are very potent antioxidants, but actually their antioxidant capacity isn't believed to be related to their beneficial effects. So we were interested to look at particular foods and how they may impact not only on vascular function, but other risk factors for cardiovascular disease. And apples is a very commonly consumed fruit within the UK, and is the second most highly consumed fruit next to bananas in our diet. They are particularly high in fibre, and it's pectin fibre, but also a lot of polyphenols, including flavonoids. So one of my PhD students, Thanasis Koutos, who's now got a lectureship in Glasgow for his PhD, was interested to look at the impact of different types of apples on the gut microbiome. And he did this initially in a batch culture, as you can see here. He looked at a number of different apples, pre-digested them, and then put them into this batch culture to see if they had any impact on the gut microbiome. And in fact, they did have quite considerable impact. This is just showing the effects on the bifidobacteria over 24 hours measured by fish. And what he found was the Canada Renata variety of apples had the greatest impact on increasing bifidobacteria in this batch culture system. These are particularly found in Italy, although you can actually buy them in the UK, and I've found them in Marks and Spencer's and Waitrose, but not all retail outlets. So we're interested to take this apple forward to see whether it had any impact within a human study. So what Thanasis did was ran a randomly controlled crossover study, looking in 40 mildly hypercholesterolemic subjects, feeding them 2 Renata Canada apples a day or an energy sugar match apple drink for 8 weeks, and then they crossed over to the other intervention. This was randomized to either of them. And what he found initially was the apples here shown in the green. There was a significant reduction in both total cholesterol and LDL cholesterol from this two apples a day. He also found that there was a greater microvascular reactivity in the apples group compared with the controls, and he measured this by laser Doppler imaging. So beneficial effects of this very small intake of apples in relation to lipids, but also vascular reactivity. We also were interested to see the impact on potential cholesterol intermediates. And so we're interested to look at bile acids here. And what he found was that secondary bile acids, this one in particular here, were associated with a greater reduction in total cholesterol after the apple consumption in women only. So in other words, those that consumed, or the women that consumed the apples, had a greater secondary bile acid production, and this was associated with total cholesterol. So how is this related? Well, there is a number of mechanisms in which this can be related to a reduction in cholesterol. So this diagrammatic shows cholesterol metabolism. If I take you first to the top bit, which is the liver, looking at cholesterol, which is converted within the liver to primary bile acids. These are secreted into the jejunum and the ileum, as they are really important in fat digestion, forming micelles to allow fat to be digested and absorbed. These bile acids are reabsorbed about 95% and recycled in this way through the liver and the gallbladder. However, there are some bacteria that have bile salt hydrolase activities. And this activity increases primary bile salts conversion to secondary bile acids. And these secondary bile acids are poorly absorbed and also are excreted to a great extent in the feces, reducing this intrahepatic circulation of cholesterol, which means that LDL cholesterol needs to be removed from the circulation with the upregulation of LDL receptors. In addition to this, these particular bacteria can produce propionate, a short-chain fatty acid, which is a metabolite that can also beneficially impact the cholesterol synthesis by reducing its activity. So therefore, this activity not only can increase the excretion of cholesterol, it also increases the uptake of cholesterol from the circulation and reduces cholesterol or bile acid production through cholesterol in the liver. And there are a number of foods that may impact on this, apples being one of them, because we know that increases bifidobacteria and other bacteria that have this bile salt hydrolase activity, and also oats. So we wanted to perform another study. This is quite complex here, but we can perform a study using the same Renata Canada apples, which you can see in the bottom row here, 2 a day. We also looked at the impact of porridge oats, and those bacteria, probiotic here, Lactobacillus reuteri, which are known for its bile salt hydrolase activities. And we compared with this control diet, which was cornflakes. 261 healthy men and women were randomly allocated to this parallel study, and the intervention was for an 8-week period. And what we looked at here was, first of all, its impact on LDL cholesterol. And we found that, as we found in the study by Thanasis, where we looked at mildly hypercholesterolemic subjects, there was a reduction in LDL cholesterol after the apples, but not after the other interventions here. This didn't reach statistical significance, but the significant reduction in serum apolipoprotein B, which is the apolipoprotein associated with LDL cholesterol in our circulation. So we found a similar effect of a beneficial impact of these apples on circulating LDL cholesterol. However, in this particular study in healthy individuals, we found no effect on other cardiovascular biomarkers or in fact on the gut bacteria or bile acids. And this could be because of perhaps the length of time of intervention, the fact that these were more healthy individuals, or perhaps other differences between the studies. So just to conclude that part of my talk, we've shown that flavonoids have a beneficial effect on cardiovascular risk. This can be modulating the vascular function, but also has impacts potentially on the gut microbiota and also on cholesterol metabolism. Another important component of fruits and vegetables, particularly green leafy and root vegetables, is that of dietary nitrates. And there has been a number of studies that have beneficially shown that higher amounts of nitrate consumption through vegetables have a beneficial effect on cardiovascular mortality. And you can see here the dark dotted line or the black dotted line is the lowest quartile of nitrate consumption through vegetables, whereas the higher quartiles you can see have a beneficial effect on survival rates. So we were interested to look at the beneficial effects of dietary nitrates on blood pressure and vascular function as markers of cardiovascular risk. This is some data from a PhD student of mine, Hayat, who completed her PhD just last year. And what she did is she looked at the National Diet Nutrition Survey for years 1 to 8. And she looked at the consumption of nitrate-rich vegetables, but also the water consumption, which contains a lot of nitrate in it. And she had to go back to the water authorities to determine the nitrate content of the water of the individuals of the NDNS. So it was no mean feat, and she did a great job here. And what she found was in those that had lower amounts of vegetable and water nitrate had higher blood pressure, this is systolic blood pressure, than those in the highest quartile, quartile 4, that had lower blood pressure. She also found that nitrate, or particularly nitrite, from processed meat, which is added to meats as a preservatives, was associated in the reciprocal relationship. So in other words, those that had high amounts of processed nitrite meat, or meats that contain nitrite, had a higher blood pressure than those that had lower amounts of processed meat. And you can also see the comparison of the blood pressure in those that had lower fruit and veg, had a higher blood pressure than those that had lower processed meat. So you can see here that dietary nitrate, particularly from a vegetable source or water source, seem to have beneficial effects on blood pressure or associations with blood pressure, but that from nitrate from processed meat did not. And this is interesting in itself, and it may be that processed meat has higher proteins and that may interact with the impact of nitrite itself, but it's unclear why there is this difference. We're interested in looking at the benefits of nitrate from vegetables on vascular function and blood pressure. This was a study performed a number of years ago by a PhD student of mine, Dita Hobbs. And what she was interested in looking at is the increasing doses of nitrate from beetroot juice of 100, 250 and 500 mils of beetroot juice that had increasing quantities of nitrate at the top of the table here. And also the purple color in beetroot is actually from betalains, not flavonoids. And also they had high amounts of betalains. And what we found with her studies, if you look at the postprandial systolic blood pressure, so this is looking over a 6-hour period, how much blood pressure change, you can see there's a linear relationship, a dose-response linear relationship with the highest doses of beetroot juice or nitrate having the greatest impact. With diastolic blood pressure, we also found a significant reduction compared with our water control, but it seemed to plateau at 100 grams of beetroot juice. Unfortunately, as a population, we are resistant to eating fruits and vegetables. So we're interested to try and put this nitrate in a food that is consumed by the vast majority of the population. And we chose bread. 96% of us in the UK purchase bread on a weekly basis. So does beetroot within bread have a beneficial effect? So this is another study that Zita performed. And what she looked at is in 14 healthy non-smoking men. She randomly assigned them to eat either 200 grams of normal white bread, 200 grams of white beetroot bread, so in other words, made with white flour, but also supplemented with white beetroot that didn't have the betalain, the purple color. And then the same quantity, 200 grams of red beetroot bread with matching nitrate concentrations within the different beetroot breads, but one containing betalains and one not. What was the impact of this on postprandial diastolic blood pressure over a 4-hour period? You can see here the control bread had no impact. The white beetroot and the red beetroot bread had very similar hypertensive effects, so lowering postprandial blood pressure. And because both beetroot breads had the similar effect, we could then conclude that the betalains themselves, the purple color, didn't seem to have any effect. And it was actually the nitrate that had these beneficial effects. We also measured the total nitric oxide over the postprandial period. And you can see here that up to 4 hours, there was an increase for both the white and purple beetroot breads. And then we took another sample at 24 hours, and you can see that most of it had returned to the levels of the control bread. So you can see here that it looks to be the nitrate that's having this beneficial effect from the vegetables on blood pressure. Dita then went on to do another human study in which oh no, so, beg your pardon, this is the same human study from previously. And this is the data from the postprandial incremental area under the curve for vascular reactivity. This is using laser Doppler imaging. And this is a non-endothelial dependent microvascular reactivity. And what she found was both the white bread and also the beetroot bread. Both the white bread had no effect on vascular function, but the beetroot bread had a beneficial effect over 6 hours on vascular function. And she found that there was, in fact, a significant increase in the plasma nitrate concentration. So we have found here that nitrates seem to have a beneficial effect on blood pressure and vascular reactivity. And this has been shown in a meta-analysis of randomly controlled trials here, published here by Jackson. And also there's beneficial effects on platelet aggregation. So nitrate, dietary nitrate seems to have benefits. Again, we were interested to determine whether there's any difference in the responsiveness to nitrates with different gene polymorphisms. We returned to the same ENOS SNP here that we looked at when we looked at the impact on flavonoid response. And here I've plotted the control bread in white and the beetroot bread in grey for the T carriers. So this is the risk carriers of this ENOS polymorphism. And they had, after the beetroot bread, you can see that both of them had a significant reduction in a systolic and diastolic blood pressure. However, if you look at the wild type variant, you see that there was not a benefit of beetroot bread in relation to vascular function. So the opposite way round in relation to the flavonoids. So the risk variant benefited from nitrates in relation to vascular function, and the wild types didn't, whereas in the flavonoids, it was the other way round. And when we looked in these two different genotype groups to the incremental area under the curve for plasma nitric oxide, we found it was actually the T carriers that the risk variant that had the higher level compared with the wild type. So again, this is a very small study and it certainly does need to be repeated, but it does indicate that certain people might respond differently to different types of fruits and vegetables in relation to benefit to vascular function and cardiovascular risk. So it may be that we need to more personalize our advice so that those that would benefit get the right advice. So nitrates have a number of potential mechanisms of action, and I just want to very briefly go through the metabolism of nitrates. So nitrates in foods are consumed, they go down to the stomach, and then they're absorbed into the circulation, and actually majority are resecreted into the oral cavity via the saliva. And this is because oral bacteria, as you can see them here in green, nitrate reducing bacteria will reduce the nitrate to nitrite. That nitrite, after being metabolized in the oral cavity, will go to the stomach. It's then absorbed into the circulation as nitrite, or there's conversion by other bacteria found in the stomach from nitrite to nitric oxide, which can have benefits in relation to the mucosa of the stomach, but also can then absorb into the circulation. The nitrite can then be metabolized further to nitric oxides, which can then have an impact on vascular reactivity and relaxation. Nitrite and nitrate can also be excreted in the urine, and we use this as a biomarker of intake. So we were particularly interested to look at the benefits of, or the potential impact of the bacteria within the oral cavity on the impact of dietary nitrates on reactivity, vascular reactivity. And a number of people have shown that if you actually eliminate bacteria from the oral cavity, you don't get the benefits of the nitrate. So we performed a study to try and identify the types of bacteria that may be associated with reduction of nitrate to nitrite and where they were in the cavity. So Hayat, my PhD student, did a study where she recruited 20 people, 10 male, 10 female, individuals, and she asked them to rinse their mouth out with 10 mils of either water, of either beetroot juice, or the Corcidil mouthwash, which will eliminate all bacteria from the mouth, and then after that, the 10 mils of the beetroot juice. And interested to see what bacteria that she could, or the nitrate, first of all, in the saliva. And what she found after the water, there was a certain amount of salivary nitrite. After the beetroot juice, you can see a very significant increase in salivary nitrite, and that's the reduction, we think, from the nitrate from the beetroot juice to nitrite via the oral bacteria. But after the mouthwash here, you can see that salivary nitrite was completely eliminated, so there was no conversion. She then looked at the impact of these treatments on bacteria, and she found here within the oral cavity different number of species, and you can see here she's mapped them onto different areas of the oral cavity. So after the water, she isolated a number of different species here, with the greatest amount here on the tongue. After the beetroot juice, this increased the nitrate reducing bacteria quite considerably here, shown in purple. But after the Corcidil, there was complete elimination of any nitrate-reducing bacteria within the mouth. Again, supporting the theory that the oral bacteria are particularly important for the beneficial effect of nitrate. So although killing bacteria within your mouth may be good for oral hygiene, it may not be beneficial for beneficial effects of dietary nitrate on cardiovascular risk. And what she did find was not only there was different bacteria within the different areas of the cavity, but the majority of them were on between the mid and the rear of the tongue. So we know, and many people have shown, that inorganic or dietary nitrate can impact beneficially on blood pressure reduction in both healthy and hypertensive populations, and has a beneficial effect on vascular function. And also that there is a differential and possibly more beneficial effect of taking your nitrates from dietary sources such as beetroot or green leafy vegetables such as spinach, rather than organic nitrates or nitrites that have been used clinically for a number of years. So, for example, if you take nitrites clinically, then these can have a negative impact on your endothelial function through the production of reactive oxygen species. However, this has not been shown when inorganic nitrates are taken in the form of vegetables. Further, if you take organic nitrites, then what happens is you have a very rapid onset of blood pressure reduction. And this can lead in some stages to hypertension that can be quite dangerous. Whereas dietary nitrates have a much slower onset, and therefore you don't get this very quick hypertensive effect. In addition to this, in the clinical nitrites, there are limitations by the development of a tolerance, which ameliorates the blood pressure lowering effects of these, whereas the nitrates in the beetroot have a beneficial, shorter, longer term effect. I can't seem to go back, but I will say my conclusion. So in conclusion, I'd like to say that I've shown you evidence that fruits and vegetables and plant-based diets are particularly important for general health, but particularly in relation to cardiovascular risk reduction, showing you mechanisms of perhaps how different types of fruits and vegetables may have differential effects, particularly concentration on flavonoids that have beneficial effects on blood pressure and vascular function, and also nitrates on blood pressure. But there may be some of us that respond better to flavonoids and nitrates, and this is something that we want to investigate further. So I'd like to just end there. I'd like to thank my research team here at the Hugh Sinclair Unit of Human Nutrition for all their hard work. And I'd just like to thank you for your attention.
Thank you very much, Julie. So I was just trying to get my computer to switch back on again on the video and on the microphone. So yeah, thanks ever so much. And I'll call for questions now, either in the chat or people can put their hands up, and I will endeavour to see them. I'm just going to ask the first question while people are thinking there. So you were saying there that there's some resistance, particularly perhaps in the UK, to eating plant foods or eating more fruits and vegetables. Is it particularly important for any one age group to do that? Is there an age after which there's no point? Or is it, very important for the young more than as one ages? Is there any information about whether or not changes in risk occur at different ages, I guess?
Thanks, Ian. It's a really interesting question. Yeah, I mean, I don't know of much data to see whether it's particularly important in certain age groups. I would say, obviously, to develop dietary habits, it's important to get our children to start eating fruits and vegetables, and hopefully they will take that through their life course and influence their children and as they go on. And of course, nutrition is particularly important in preventing disease. So if you can prevent cardiovascular disease and other diseases, then it's much better than trying to treat them. I think that so far as intake, that our intake of fruits and vegetables can reduce as we get older, and it may be a reflection of perhaps dentition, with fruits and vegetables, particularly fresh ones, more difficult to chew, and also the impact of a varied diet is not necessarily in the very older people followed as it perhaps should be. So, I mean, I would say that perhaps in all age groups, trying to instill this sort of dietary intake in the young if we can.
Yeah, thank you. Any other questions? I'm just trying to, a hand is up from Anne-Marie. Anne-Marie.
Yeah, thanks a million, Julie. That was fantastic. Can I ask about whether there's any, I mean, the fruit and veg recommendation in the UK is quite old. We've been living with the 5 a day for probably decades now. And do you think there's any kind of push at a policy level to kind of be more specific and refine our current recommendations? I know for brain health, things like green leafy vegetables and berry fruits are certainly most beneficial when you look at fruits and vegetables. And you've shown very beautifully there that when you're talking about cardiovascular health, apples, high nitrate fruits and veg may be more beneficial. Is there any push at a policy level to get more specific, do you think?
Yeah, I mean, that's a really, really interesting, important question. I mean, I think, in the day when we did flavors, Anne-Marie, which was a number of years ago, 10 years ago, I think the FSA, as was then, was really interested in trying to determine whether there are any specific fruits and vegetables that may be more important than others. And I think this is something that we have now more evidence about. And I think that, as you say, many people are looking at flavonoids and fruits, as you say, fruits and vegetables that are rich in those in relation to cognitive function as well as cardiovascular risk. And I think that, there may be opportunity to change our recommendations to much more or give clearer guidance to people what sorts of fruits and vegetables should be consumed. As I'm aware, in the UK, that's not something that's on the agenda at the moment, but it is something I think is important. And in relation to flavanols, I know that the Cosmos study, the group there, I mean, it was funded by Mars particularly, but I think a lot of people, particularly in the US, are really pushing for a dietary recommendation for flavanols in itself. And I would hope that would be through diet rather than supplements, but they are really pushing for that with this evidence of hard clinical outcomes of flavanols. Then you never know, there may be maybe traction in that.
Yeah, Thanks, Julie. I think the evidence for flavonoids is pretty convincing now.
Yeah.
I think the time is now, really. Thank you.
So there's a question in the chat from Peter, which I've now lost. Here it comes again. How does the strength of the association between fruit and veg intake and cardioprotection compare against other lifestyle behaviors like weight loss, physical activity? And how do they compare against pharmacological intervention is the question.
Yeah, great question again. I don't know if there's been any absolute comparison between these. I think that with any guidance, dietary guidance, obviously we should say that, it should be taken in the context of a balanced diet, and that, in isolation, fruits and vegetables are good. And I think the more, probably the better, up to a certain extent. And I think the 5 a day that we say in the UK and generally around Europe, it could be pragmatic in a way. Our intake is very low, but I think higher amounts could be better. Although there is evidence that 5 a day, or lower than 5 a day, has detriment. I think that there are a number of pharmacological interventions, such as statins, that have a very beneficial and significant reduction, for example, in cholesterol. And it's not as though I would recommend fruits and vegetables over and above that, or instead of something like a statin. But I think the mechanisms by which they act could be complementary. So, for example, the flavonoids and the nitrates work in different ways. And I think that if you have a combination of those, then that may have potentially synergistic effects, and therefore could complement other therapies that may be given to an individual. But also, I think in the context of increasing physical activity, for sure, and also weight loss is important. But it can also be helpful in those ways. I mean, in relation to weight loss, if you have a lower energy dense diet, which often comes from higher fruits and vegetables, that can help with weight loss.
Yeah, thanks, Julia. I was also going to ask, with your study where you looked at oats and probiotics and apples, whether you had looked any further on synergy between those. So if you have apples and oats, or if you have apples and prebiotics, whether that gives you, more than an additive effect, or.
Yeah, we haven't, because it was a parallel study. So they each of the groups had just the individual intervention, but it's a really important point. And we are really interested in looking at nitrates and flavonoids together. And to see whether there is synergy there. There is some evidence that actually it may not be synergistic. It may even have detriments at the same time in the stomach. But that, I think, is really interesting, because to give guidance to someone even about when you're to take fruits and vegetables may also be interesting, important. But I think these particular compounds within fruits and vegetables, do work by different mechanisms, could work synergistically, but then equally could work against each other in some respects.
Yeah, thank you. So in the chat, Sue Fairweather Tate asks, who's responsible for developing nutrition strategy in the UK?
Nutrition strategy. So in relation, so as you know, obviously, Scientific Advisory Committee for Nutrition is the government advisory committee that looks at risk of and benefits of diets and dietary components and nutrients within the UK population, and will advise government on policy development. The government doesn't have to listen to our advice, and in relation to folic acid, has taken 20 years for them to actually acknowledge that perhaps increasing folic acid within our diet and fortification is of benefit. So it does. In SACM, we look at risk. But we don't just look at assess the evidence. We don't look at risk management, and management's done by OHID at the moment. So I think it is important that we have horizon scanning meetings at SACM, and also other people can write in if they have any particular importance or interest in it, how we should prioritize our advice, and we can always then take that and discuss that amongst ourselves, and therefore decide what is a priority for the UK dietary policy.
Thanks, Judy. There's another question in the chat from Yang that says, does beetroot contain a lot of nitrate? How do you determine the concentration? And whilst I may have missed it, but how do you incorporate beets into bread, it says. So yeah, three questions there.
Yeah, so root vegetables generally have a lot of nitrate in them. The amounts that they contain does depend a lot on the soil. So a lot of people top dress with nitrogen-containing fertilizers. And of course, if you've got a nitrate-rich soil, then the root will take it up. So you can increase it depending on that. You can measure quite easily nitrate concentrations. So we can do it by HPLC, for example. I'm sorry, I forgot the third one now. Incorporate into bread. So we were interested to see whether it had any differential effect when you processed it and put it into bread. And so my student who was really interested in baking, what he did is he not only put beetroot juice into the bread, but also kind of chopped up, I said that's a very bad way of describing it, but it chopped up beetroot. So there was beetroot pieces, so he cooked the beetroot, then he chopped it up quite finely, incorporated that into the bread as well as beetroot juice. And from the results, you can get quite a lot of nitrate into bread by that means. And it rose, and we did a lot of sensory work on it, which I didn't show you. And it was actually enjoyed by people. The taste was enjoyed. It had a slightly earthier taste, as you might expect, than other breads. The colour was unusual. Children quite liked the colour. And so it was accepted, and it was consumed, and there was quite a lot of positive responses when we did sensory analysis from it.
Thank you, Julie. There's another question. Oh, sorry, it keeps flipping backwards and forwards on my screen from Peter that says, every time someone types something in, it flips to another point. It says, the nitrite biology is fascinating. The differential effect on health between nitrates in fresh veg and those in processed foods suggests different metabolites are produced. Has that been looked at?
Yeah, that is really important, and it does seem contraindicative, in relation to, we're talking about nitrites that are generally in processed foods, and of course there's this big interest in ultra processed foods. We won't necessarily go there now, but they have seemed to have a detrimental effect, and Hayat showed with the NDNS, but that was just associations. But there is a lot of evidence that processed meats, particularly red meats, are associated, for example, with some cancers, colon cancers. And it could be that you have nitrates with protein. You might get nitrosa compounds produced in the gut, which can then have damage on the gut wall and increase carcinogens and that mechanism. We don't know about nitrates in fruits and vegetables. Generally, it's low protein foods. I don't think there's been done much work on looking at fruits and vegetables containing nitrate and the gut microbiome. And I think that's really important to ensure that you don't get these detrimental compounds being produced there. I haven't seen any evidence that there are, but I think this is something we do need to look into. And of course, we do have an upper limit of nitrate that we're meant to consume in a day. And actually, these studies that we've done and others is above that. So we're actually above the quantity that's meant to be a maximum. And that's generally because of the impact on babies, where you can get the blue baby syndrome that has detrimental effects. But in the general population, I feel as though we need to revise that upper limit, because I think the evidence for detriment on the general population, I don't think there's strong evidence about that.
Thank you, Jill. I just wanted to read out from the chat that both Jonathan and Sue Fairweather Tate echo your comments about how difficult it is to get government sometimes to listen. So both of them absolutely echoed your comments on that. Any other questions from the audience, either in the chat or by people putting their hands up? I can't see any, in which case, again, thank you very much, Julie, for a fantastic presentation. And even more thanks for coming back and giving us that presentation after all of our problems last time. So that's absolutely appreciated. And yeah, again, thank you for such a fantastic presentation, and for everybody in the audience for the questions that they've been asking. So yeah, thank you very much indeed.
Thank you very much, Ian and Anne-Marie, and thanks to the audience. And it's a great pleasure. I'm delighted to be able to present to you.
Thank you.
Thank you very much.